Okay, now today we are going to talk about allosteric regulation of enzymes. Let's walk through this lesson in a very short time. I'm assuring that you will understand each and every theory fact about allosteric regulation of enzymes. When we consider normal conditions, normally enzyme regulation mechanism occurs through non-competitive reversible regulatory molecules. That's the normal procedure. So, when considering about allosteric regulation, we have to understand that allosteric regulation is based on two types. They are allosteric regulation of activation and allosteric regulation of inhibition. So, allosteric molecules may be of two types. They may be activation molecules or inhibitory molecules. Before going into details, let's understand what the meaning of this allosteric word is. Now, normally an enzyme regulation mechanism can occur through two methods. I will depict a normal enzyme. This is just a typical enzymatic diagram I am drawing. So let's consider this as the active site. I'll indicate with a star. This is the active site, and if the regulatory molecule binds to the active site, this is a regulatory molecule. If the regulatory molecule binds to the active site, we call it as a competitive regulation. If the regulatory molecule binds to any other site other than the active site, then that is an allosteric mechanism. So, simply allosteric regulation is regulation occurring through any other site other than the active site. So, when considering any other site other than the active site, it simply means that the active site will remain free even when the allosteric regulation occurs, so that the active site is available for the substrate molecules. When allosteric regulatory molecule, let's consider an allosteric activator, the allosteric activator comes and binds to the allosteric site, the active site will remain free and open, so that this active site will be available for substrate molecules for binding. According to our A-level syllabus, we have three subtopics under the topic allosteric regulation of enzymes. So, we will discuss one by one and understand each and every theory point so that we can easily face our A-level examination when questions are questioned regarding the following topic: allosteric activation and inhibition. When talking about allosteric activation and inhibition, the first fact that we should remember is allosteric regulation occurs for enzymes with two or more subunits. We have to by heart this term, two or more, two or more subunits, because in several past papers we can see that they have changed simple words of these phrase and questions, so that the student, when facing the examination with the exam stress, he will quickly misinterpret the question. So, by chance they may ask one or more subunit, then it's a wrong statement. It is very clearly mentioned in our resource book that for allosteric regulation to occur, there must be two or more subunits in the enzyme complex. So let us consider an enzyme complex with four subunits. Now this is, I am just typically depicting a structure of an enzyme. This is not the way a real enzyme will be seen. And let me depict the active sites of each enzyme subunit. Now these are our active sites, and these are the active sites, and we know any other site other than the active site will be termed as an allosteric site. We can even term them as regulatory sites. So here I am marking several allosteric sites. Now, according to our first subtopic, allosteric activation and inhibition. So you can see even in the topic there are two parts: activation and inhibition. So now let's consider this enzyme complex that I have drawn. This enzyme complex is oscillating between two forms as the stable active form and inactive form. Now this is just a graphical representation I am reminding you this thing that because you may misunderstand that this is how a real enzyme is seen. No, this is just an imaginary diagram we are drawing to understand the process. Now this is the active form. Let's mention active form. Then this is the inactive form. You can see in the inactive form, the active sites are not properly arranged. The active sites are disintegrated. Now, for in between these two, active form and inactive form, allosteric regulation occurs to stabilize one form. So let's see, there are two types of allosteric molecules I mentioned earlier, allosteric activators and allosteric inhibitors. So if an allosteric activator binds to these oscillating complex, now let's depict our allosteric activator like this. This is an allosteric activator. Now it comes and binds to the allosteric site. You can see the active sites are not barriered by the allosteric activator. So still the active sites are available for the substrate molecules. And when the activator molecule binds to the allosteric site, then the active form stabilizes. The oscillation process stops and the active form will stabilize. Now the active sites are stable, so the active sites are open. Now substrate molecules, let's consider this as a substrate molecule. Substrate molecules can bind to the active sites. Now the speciality of this allosteric regulation is when an allosteric activator or inhibitor binds to one allosteric site, this regulatory signal will pass along onto all its subunits. Then let's consider this example. Allosteric regulatory molecule binds to the allosteric site to one allosteric site, then it passes signals to all the subunits. What I am trying to explain is that you do not have to bind allosteric molecules to all allosteric sites for the complete enzyme to be regulated. By even binding one allosteric molecule to the allosteric site, you can pass along the regulatory mechanism onto all the subunits. Now let's consider the inactive form. As I mentioned earlier, for the active form to be stabilized, an allosteric activator should bind to an allosteric site. Then for the inactive form to stabilize, an allosteric inhibitor should bind. So let's depict the allosteric inhibitor as follows. Now here I have drawn an allosteric inhibitor. Okay. When allosteric inhibitor binds to one allosteric site, then the inhibitory mechanism passes along to all the subunits, making all the subunits stable in its inactive form. Then when the inhibitor binds, the inactive form stabilizes, stopping the oscillation process. Now let's move along to our example from allosteric activation and inhibition. I suggest that you should take your notes when the video is going along, because this section, allosteric regulation, has been questioned thoroughly each and every year in our past papers. ATP and ADP are two types of allosteric molecules. Under this section, we will be discussing about both of them, and I will remember when you proceed along your second unit, you will be realizing and studying more and more about ATP, which is adenosine triphosphate. For now, let's talk about ADP. ADP or adenosine diphosphate is a type of allosteric activator. It binds to the enzyme which catalyzes catabolism reaction and synthesizes more and more ATP. So ADP binds to the enzyme, not to the active site, but to another site other than the active site, acting as an allosteric activator for the synthesis of ATP. Then when excess ATP is present, ATP binds to that same enzyme and ATP acts as an allosteric inhibitor. Because ATP binds to the enzyme, hence the enzyme inactive form is stabilized, resulting in stopping the synthesis of ATP. You will be understanding about this ADP and ATP mechanism in our third subunit, feedback inhibition. For now, under allosteric activation and inhibition, this is the example you should be able to understand when answering MCQs and structures mainly. Now let's proceed to our second subtopic, cooperativity. Cooperativity is another type of allosteric regulation. Here also we talk about an enzyme complex of two or more subunits. That doesn't change, that's a theory fact. So let's consider another enzyme with four subunits. I'm choosing four subunits for the ease of studying. Now let's see, you can see I have chosen an enzyme with four subunits. In cooperativity, what happens is we do not bind an allosteric regulatory molecule to a regulatory site or an allosteric site. Here we bind one substrate to the active site itself. I said we should keep the active sites free to the substrates. Now here we do not use an allosteric regulatory molecule. We use the substrate molecule itself and we bind the substrate molecule to one active site. Now we do not have to bind substrates to all four active sites. Let me depict a substrate molecule like this, and I have bound the substrate molecule to one active site. In cooperativity, what occurs is the binding of one substrate molecule to one active site of a subunit. This provides signals to other subunits. It provides signals to other subunits, which increases the affinity of the other unbound subunits. Increases the affinity of the subunits which the substrate are still not bound, making it more efficient for binding of the substrate molecule. So, in other terms, binding of one substrate molecules increases the affinity of other active sites for the quick binding of the substrate molecules. In our syllabus, we have an example. We should keep in mind this example is not about an enzyme. We just use this example because it's very easy to understand and it correctly depicts the process of cooperativity. We consider the hemoglobin protein molecule. It's not an enzyme. I am remembering you again. This could be termed in A-level questions. Many students have misunderstood this point. Hemoglobin is not an enzyme. It's very clearly stated in the resource book. Hemoglobin, not an enzyme, in brackets. So we should remember that point, and we will discuss about the example. Hemoglobin is also a molecule with four subunits. We know the subunits, we term them as alpha globin and beta globin. We learn about this hemoglobin in the second unit in chemical basis of life under proteins subtopic. I think you remember it. We term two type. We understand about two types of subunits as alpha globin and beta globin in hemoglobin molecule. Similar to this cooperativity process, when the subunits of hemoglobin are waiting for oxygen, the heme groups of the subunits are waiting for oxygen to bind. Then in this moment, when one molecule of oxygen binds with the heme group of one subunit, this increases the affinity of other subunits to the binding of molecular oxygen. I think the fact is clear. I will remember you again. This is not an enzyme. We just use the example hemoglobin because it's very closely identical and very similar to the cooperativity process. I think you have 100% guaranteed clear about cooperativity. Now let's talk about feedback inhibition, which is the final topic of allosteric regulation. Feedback inhibition. In feedback inhibition, a metabolic pathway is stopped by inhibitory binding of the end products to the enzyme. The special factor is the end products itself bind to the enzyme to inhibit the metabolic pathway that these end products were created. The speciality of this fact is this limits the excess production of this limits the excessive production of end products, which will cause to reduce or eliminate the wastage of end products. So this is a very advantageous process. When we talk about feedback inhibition, we have to talk about the example we talked in our first subtopic about ATP and ADP. ADP is an allosteric activator. ADP binds to the enzyme to produce more and more ATP through the metabolic process. When ATP is produced in excess, ATP is the end product. When ATP is produced in excess, this ATP itself binds to the enzyme to inhibit the production of ATP. It's like this. Now the process is production of ATP from ADP, and here is the enzyme. Let's resemble it in this manner. When ATP is produced in excess, this ATP itself binds to the enzyme and inhibits the metabolic pathway of producing the end product. I will mention this point very clearly again. This is an advantageous process because this limits and eliminates the excess wastage of end products. So in a very short period of time, we have completed discussing every single fact about allosteric regulation of enzymes within the frame of your A-level syllabus. I think you could grasp all the theory points, and I prefer if you made your own theory note, including all the MCQ points I discussed throughout the video. Now you may use your Pesuru book to do the past papers related to allosteric regulation. Allosteric regulation is a part that has been questioned throughout A-level past papers every year. So, I mean, it's an asset to fix this theory section, because you will be assured by MCQ, and also there is a higher probability of receiving an A-level essay question, or at least as part of a structured question, mainly the first structure or the second structure, a subsection of the structure from allosteric regulation. Because I will mention it again, allosteric regulation is a theory section that has been continuously and repeatedly questioned each and every year. So with that, I think you could grasp all the theory points. If you have any doubts, I suggest you to rewind the video and study with me again. So let's meet in our next video with another interesting biology topic.